Welcome everyone. Today we are going to learn about sodium glucose Symporter. But before I begin, at first we have to understand four important points. The first point is downhill movement. Second point is uphill movement. Third point is chemical concentration gradient. And fourth point is membrane potential or electric potential. In short, we could say voltage difference across the plasma membrane. So these are the 4 points we have to understand at first to understand the mechanism of sodium glucose Symporter. At first, let me talk about these two movements, downhill movement and uphill movement. Suppose this is an example, this is the example of hill. We know if someone try to come down from a hill, it requires less energy, less effort, it feels easy. So in case of downhill movement, it is easy. But when someone try to climb up the hill, so we call uphill movement, it requires more energy and more effort. Okay, so the same is true for the movement of molecules also. Molecules also perform this downhill and uphill movement, and in case of downhill movement, molecules move from higher to lower concentration, and during this transport no energy is required because this is thermodynamically favorable. That's why this movement is called passive transport. So we have to understand these points at first. This is called passive transport. In case of uphill movement, molecules move from lower to higher concentration. And during this movement, energy input is required because this process is thermodynamically unfavorable. That's why this process is called active transport, because here energy input is important. Now, in my previous video, I have mentioned different types of passive transport. I'm also mentioning here briefly. So passive transport in case of cells are performed during simple diffusion, by uniporter, and by channels. Now, in case of simple diffusion, no transport proteins is required. Molecules move directly through the plasma membrane or across the plasma membrane. So I'm not considering simple diffusion, but here also molecules move from higher to lower concentration. That's why I have categorized these three into the passive transport. But no transport proteins required for simple diffusions. In case of uniporter, these are transport proteins. Uni means one kind of molecules actually transported through this uniporter. In my previous video, I have actually discussed about one example of uniporters is glut protein. Glut proteins in most cells that import glucose contains glut protein. So through the glut protein, glucose import occur. That means glucose comes inside the cell. And through the channels, ions and waters can move from higher to lower concentration. So this is examples of passive transport. Now let's take an example of active transport. It will help us to understand the Symporter also, this process. Transport. Okay. So in case of active transport, there is one mechanism called coupled transport or co-transport. Now in this case, two molecules move simultaneously. No one molecule can move either. It requires the help of the other. That's why it's called coupled. It is obligatory. Now there is two types of co-transporter, coupled transporter present. One is called Symporter, and another is called antiporter. We are learning today this category of transport proteins, that is Symporter. In case of Symporter, two molecules move in the same directions. In case of antiporter, two molecules move in opposite directions. Now there is another two types of active transport occur, that is by ATP driven pump. Here ATP, adenosine triphosphate, there is a 3-phosphate group attached with phosphoanhydride bond. The terminal phosphate group is actually break down to make it ADP, and the release of inorganic phosphate generates energy. That gives this transporter the energy input. Now how this coupled transporter gets the energy, I will explain here in this video. Now in case of ATP driven pump, ATP breakdown generates the energy to move molecules from lower to higher concentration, because these molecules are moving against their concentration gradient. So this chemical concentration gradient. Now another type is light driven pump. I have made a separate video on light driven pump. Example is bacteriorhodopsins. Now here the light, solar energy acts as outside energy or output energy. So this is all about the downhill movement and uphill movement. I think you have understood. We have to understand these two movements at first, because in case of Symporter and antiporter, both these cases, two movement occurs. Okay, two movement occurs. Why it is called under the active transport? Because one molecule will show the uphill movement and another will show the downhill movement. The movement of downhill movement actually generates the energy to run the uphill movement. I will explain it, but before that we have to understand this third and fourth point, that is membrane electric potential, membrane or electric potential, or chemical concentration gradient. So let me explain. At first I will remove this. To understand what is chemical concentration gradient and membrane potential or electric potential, I have to draw plasma membrane. Suppose this is the lipid bilayer, the plasma membrane. Okay, and this is the outside of the plasma membrane, that means outside of the cell, and this is the cytosol. So we are actually learning about 2 points. One is chemical concentration gradient and another is membrane or electric potential, that means the voltage difference in between the plasma membrane or the lipid bilayer. Now at first I will talk about this chemical concentration gradient, this factor. Molecules that does not have charge, that means for uncharged molecules, this chemical concentration gradient acts as the driving force, of the movement. But those molecules contain charge, like here uncharged molecules, examples like glucose, fructose, etc. And in case of charged molecules, it could be ATP, amino acids, etc. For charged molecules or ions, then both the chemical concentration gradient and the membrane or electric potential together work. So for charge and ions, these two factors work, and togetherly we call this force, we call it electrochemical gradient. Electrochemical gradient. in case of ions, we specifically call this force ion motive force. The same terminology we use for ions, specifically ion motive force. So these two factors actually generate the driving force. So in case of uncharged molecules like glucose, this chemical concentration gradient is the only driving force. But in case of charged molecules like ATP, amino acids, and also for ions, both the chemical concentration gradient and the membrane potential or electric potential together we call electrochemical gradient, or in case of ions we call it ion motive force, together they work. Now, at first I will talk about the example of chemical concentration gradient by glucose. See, in most cases, glucose is present higher in concentration outside. So glucose concentration is outside than the inside. Okay, like in the blood, the glucose concentration is high than most of the cells, or in the extracellular body fluids, the glucose concentration is higher outside than inside. Now, we can draw the chemical concentration gradient as triangle. We can draw it by triangle. The base of the triangle indicates that here the concentration of the molecule is higher, and the peak point indicates that here the concentration of the chemical is lower. So, as because glucose present outside is higher, so we can draw the triangle like this. The base will be outside and the point will be inside, because here the concentration of glucose is low. So by this way, we actually represent the concentration gradient or chemical concentration gradient. In case of glucose, it will be like this, inverted triangle. Suppose this is the GLUT proteins. I have already mentioned that through the GLUT proteins, glucose actually moves from higher to lower concentration. So here, glucose will move from higher to lower concentrations. And here, this chemical concentration gradient gives the driving force. Let's put some number. So suppose this driving force of chemical concentration gradient is 2 for glucose. Now, in case of, let's now take example of ions, then we will understand both this chemical concentration gradient and membrane electric potential together we call electrochemical gradient. Let's take an example of ions. But before that, we have to understand two other proteins that creates this membrane or electric potential. The first protein here is called sodium potassium pump. This protein is called sodium potassium pump. This is example of ATP driven pump. Here ATP breaks down and 2 potassium actually enters the cells and 3 sodium exit the cell. Okay, this is not antipotar, this is a pump. By breaking down 1 ATP, 3 sodium moves out and 2 potassium comes in. See here that during each cycle of ATP breakdown, as because more positive ions are moving outside and less positive ions are moving inside, a charge separation starts to occur. So more positive ions will be outside. All over the membrane, more positive charge will present outside than inside. So inside will be negatively charged. Okay, we have to understand this. This is because this is electrogenic in nature. 3 sodium comes out and 2 potassium comes in. Now, this sodium potassium pumps create ion concentration gradient or chemical concentration gradient of sodium and potassium. I am just giving the example of sodium here because I will take the example of sodium because we are learning about sodium glucose Symporter. In case of sodium, it has been found the outside concentration of sodium is 145 millimolar in case of mammalian cells, and inside is 12 millimolar for the sodium. So inside concentration of sodium is low, outside is higher. So we can draw the triangle for sodium. Let's suppose this is the sodium. So we can draw the triangle of chemical concentration gradient like this. The outside will be the base, the inside will be the point. So this is the chemical concentration gradient for sodium. Okay, and this is the glucose. See, as the sodium is actually ions, so it carries charge. So it has concentration, chemical concentration gradient, that is the chemical concentration generated by sodium potassium pump. Also, it has a membrane effects of membrane or electric potential that is generating here by the sodium potassium pump. Not only that, another protein helps in the establishment of membrane or electric potential, that is called a channel called potassium leaky channel. So this is called non-gated potassium leaky channel. In resting conditions, these channels are open and these channels are present in all animal cells. That's why all animal cells has a resting membrane potential or electric potential. And what happens here, as because you know, more potassium ions enter, it actually generates a potassium concentration gradient. So potassium ions actually move out of the cell. Here, in case of potassium ions, the potassium concentration is higher inside and lower outside. So these channels actually move potassium from inside to the outside, because I have mentioned channels actually show passive transport. So potassium ions is present higher in concentration here than outside. Now what I am talking about, by this moving of positive ions by these channels, more positive ions will generate outside. More positive ions will be generated and less negative and more negative charge, because there is lack of positive ions present inside. And this generate one important point, we call it resting membrane potentials. So I can write here resting membrane potential or electric potential, that is about -70 millivolt, as because this is the inside is negatively charged, so we write it like -70 millivolt. But the resting membrane potential can vary. It can vary -20 millivolt to -200 millivolt, and it depends on the cells and the organisms. So generally it is 70 millivolt. So this is the case that more positive ions present outside than the inside. Now in case of sodium, suppose in case of sodium, the entry, suppose yes, now suppose this is the sodium glucose Symporter. In case of sodium glucose Symporter, what is happening here? See, this is sodium ions. Okay, in case of, let's suppose this is the sodium and glucose Symporter. I'm just mentioning the role of sodium here only, as because the concentration gradient, chemical concentration gradient is higher outside than inside. So sodium will move from outside to the inside, and that will give a point like I have given here, the point +2. Let's suppose I am giving point 2. But due to the presence of membrane or electric potential, because here inside is negative, more positive ions will be attracted. So positive ions will be attracted inside. That will give another point. Suppose we are giving another 2 points. So total points will be 4. See here, this is the driving force of the molecules. For molecules that don't carry any charge, we call uncharged molecules like glucose, only the chemical concentration gradient is the driving force. But for ions or molecules that has charge, I'm giving example of sodium, as the concentration gradient, it has a charge. So chemical concentration gradient that is generated by sodium potassium pump, higher concentration of sodium outside than inside, so it gives a force. I'm taking it +2, and the membrane electric potentials that is generated by this mechanism of these two proteins, that also gives the driving force for sodium ions to move inside the cells. So it is giving another +2. Let's suppose that one. So what I want to mention, that these two driving force can act together to move one ion.